So, hi everyone. I don't know if I'm supposed to begin, but I will begin now. So, my name is Agathe Puissant. I'm a final year PhD at the Natural History Museum in Paris. And I'm doing my PhD on color pattern diversification in swallowtail butterflies. And so today I'm going to talk to you about a part of my PhD that I've spent on ultraviolet color pattern diversification in this family of butterflies. So butterflies can see through their composed eyes using proteins which are called opsins, which absorb light at various wavelengths. So here are some absorption spectra of various species of butterflies. So you can see that they have opsins that absorb between 400 and 700 nanometers, which corresponds to the human visible light. But they also have opsins that absorb below the 400 nanometers marks, which corresponds to the ultraviolet or UV range. So this means that a butterfly that appears to us as being uniformly yellow will appear very differently to other butterflies, and also to some of their main vertebrate predators, which are birds, where some species can also perceive UV. So why is the vision of butterflies and their predators important? It's because it will impact the evolution of their color pattern, especially in the case of sympatry. So sympatry is when species share some part of their geographical range and frequently encounter each other, which means that they will interact. And this can have several evolutionary outcomes on their color pattern diversification. So first, because they share the same predator communities, it can lead to the convergence of their color pattern via mimicry. So mimicry is when species that can be very distantly related may display a very similar color pattern that are associated with some kind of toxicity, for example. But being in sympatry can also lead to other types of interspecific interactions, such as reproductive interferences. So reproductive interferences is when individuals from different species, usually phenotypically similar species, will try to mate together or compete for territory or mates, which will be costly in terms of times and resources, and can lead their color pattern to diverge. So we've already looked into the effect of sympatry on convergence and divergence of color pattern in swallowtail butterflies in visible light, and we found that overall there is sympatry convergence at a macroevolutionary scale, but we also found some signal for divergence, but that is restricted to closely related species and especially for males. So now what about the UV part of the color pattern that we did not take into account? So there are several selection pressures on the evolution of UV color pattern. So first, predation. So it has been shown that having a more UV reflective color pattern is linked with increased predation pressure. And in the cases of mimicry, it has been also shown that the mimicry extends onto the UV range. So the mimics are also similar in the UV. So on the other hand, UV color patterns are also involved in courtship and in mate choice. And in several species, it has been shown that females prefer to mate with the males that have a more brightly colored pattern in the UV. And in sympatry, it can lead to more complex cases where UV has been shown to be used as a cue for species recognition. So for example, here are sister species of Coleas that are found in sympatry. And you can see that they are kind of similar in their visible light color pattern. But when you look at the UV signal, the males are very different. And experiments have shown that the females use this UV signal to recognize conspecific from heterospecifics. So it's likely that this balance of natural selection by predators and sexual selection will impact differently the dorsal and the ventral side of the butterflies, which are not visible at the same time to the same species. And some previous studies have started to look into this differential evolution in another family of butterflies and found some differential evolution of eye spots in the Biciclus family. So here I'm interested in the diversification of UV color pattern at the macroevolutionary scale, and particularly the main selective pressure on the dorsal versus the ventral sides of my butterflies, and what is the effect of interspecific interaction in sympatry on the UV color pattern diversification. So to do this, I'm working with the Papilionidae family, the swallowtail butterflies, for which I have a phylogeny, so I know the phylogenetic relations between the species. And I also have a visible light photography dataset of the specimens of the Natural History Museum in Paris for more than 300 species. So, and this family is very interesting because species display a wide variation of both color pattern, but also of wing shape, as you can see on the picture. So to study the ultraviolet color patterns, I took some UV photography using a particular setup of those same specimens. And so here are some examples of UV pictures. So the whiter the pixel, the more UV reflective it is. And I managed to get 113 species in common between the visible light dataset and the ultraviolet dataset. So now a big part of my thesis has been spent to find out how can I quantify color pattern similarity between my species. And because, as I said, I'm working at a large macroevolutionary scale, species have a very wide variation of wing shape that prevent me from using the typical technique where you align the wings together and then compare the color pattern pixel by pixel. And so that's why I developed a method that is based on unsupervised machine learning to automatically quantify similarity between images. So I don't have the time to get into details for this method, but I will briefly explain how it works. So from your original images of butterfly, you produce modifications from those images by doing cropping, rotation, etc. And then during the training, the neural network will learn what are the discriminative features from modifications that come from a same image that make them similar, because they come from the same image. And what are the discriminant features from modifications that come from a different image that makes them dissimilar, because they come from a different image. So at the end of the training, what you get is a vector that represents all those discriminant features that were learned by the neural network that you can visualize on the images. So these are the redder parts of the images. And you can actually consider these vectors as coordinates in a multivariate morphospace. And so here is a projection of the learned morphospace for the visible light pictures using this method. So for the UV pictures, I actually use the same method, but this time I trained it using grayscale images so that the neural network could learn how to quantify similarity between my grayscale UV pictures. So I trained it in the same way, retrieved the vectors from my UV pictures, and in the end, I could get, in the same way, a multivariate UV morphospace. So just to sum up, for each species, I now have two multivariate traits, one corresponding to the visible light color pattern and one corresponding to the UV color pattern. So now the first thing we looked at on our data was the mean UV brightness of the wings, and we compared the dorsal and the ventral side, and also between the males and the females. So here are some examples of the pictures of the dorsal side and the corresponding distribution. So in green you have the males and in blue the females. And now here are the corresponding ventral sides and the distribution. And so what we can already see is that the ventral sides are brighter than the dorsal sides in the UV. And it was quite systematically the case in my dataset. I think I have like 3 species for which it's not the case. So already there is something different happening on the ventral and the dorsal side. Next, we calculated the evolutionary rates to compare the dorsal rates and the ventral side rates. So first, in the UV, we found that the ventral side evolves faster than the ventral sides evolve faster than the dorsal side, especially for males, where it's 3.3 times faster. And when we looked at the same thing in visible light, we found smaller differences, so no difference for the females, and a smaller difference for the males. So ventral sides seem to have a higher evolutionary rate in the UV, especially for males, and the dorsal side evolution seemed to be more constrained than the ventral side, especially in the UV. So next, we looked at the effect of interspecific interaction and to look at whether they impact ventral and dorsal side evolution differently. So to do this, we tested for the effect of sympatry on UV color pattern diversification. And we did this in the sister species in our dataset that are the more closely related species that are more likely to be impacted by this type of interactions. And so we first computed the pairwise distance between the species in the UV morphospace, and explained it by the distances, but this time in the visible light morphospace. And we do this to remove the part of the variation in the UV that is due to the variation in visible light, and so to look only at the specific signal on the UV color pattern diversification. So next we added the range overlap as a proxy for sympatry, and the phylogenetic distance to control for, because some sister species are more closely related than others. And so here are the results of the model. So the only interesting line in this table is this one. It's for the effect of range overlap on the ventral sides of males. And the coefficient is positive, which means that the more you have overlapping range, the more the males tend to have different ventral sides in the UV after taking account of the variation for the visible light. So in other words, it means that there is divergence of UV color pattern for the ventral sides of males. And this corresponds to this type of cases. So these are two species of papilio that live in the US that are kind of similar in visible light, but when you look in the UV part of the signal, they are very different. So just to discuss briefly about these results, we found brighter and more rapidly evolving UV color pattern on the ventral sides, which points to differential selection pressure on both sides. And because the dorsal side is dimmer and seems to have a more constrained evolution, we hypothesize that it's more impacted by predation pressure. And because we found divergence for the ventral side of sympatric males, it points to the UV signal being used as a supplementary cue for species recognition in sympatry by females. And so we hypothesize that the ventral side is more impacted by sexual selection. But now, mate-choice experiment would be needed to investigate a little bit more about this hypothesis. So with that, I'd like to thank you for your attention, and I'd like to thank my collaborators, and particularly my advisor with whom I did this work. So thank you very much. I think I have like 1 minute for questions. Yes?
So just a quick question. With UV photography, you can probably say whether there's presence of UV or not. But could you also quantify whether the reflectance peaks, like is there variation within like the 300 to 400?
So I think to do this, you would have to use like a multispectral, hyperspectral camera where you really have the spectra for each pixel of the images. Because for me, I just have like a sum up of the signal in the UV. Like I can't really see the variation. Like you can, I have only one sensor in my camera that tells me UV, you know. Contrary to a visible light camera where you have red, green, and blue, and you have the different variation for each sensor. Yes?
So you said you had 130 species, and I was wondering if you have an intra-species variation in the UV.
So in my study, I use mean phenotypes per species, but when I took the pictures, I took up to 4 by sex by species to make sure that there was not like a lot of intraspecific variation to check, and also to get a mean phenotype for my study. Okay, thank you.
Hi, everyone. Sorry for the delay. My name is Ava Ghezelayagh. I'm a postdoc at the University of Chicago in the lab of David Jablonski. And today I'm going to be discussing how certain bivalve taxa are moving into the deep sea using a phylogenetic framework. So when we're kind of clarifying the spatial origins of diversity, you can't even see that slide from here. When you're clarifying the spatial origins of diversification and understanding patterns of global biodiversity, in general, we're thinking of things in 2 dimensions, but for some taxa, it's important to think about things in 3 dimensions, right? On land, that might be a matter of altitude, and then in the oceans or in lakes, right, we're considering bathymetric depth, right? And as you're kind of moving down into deeper waters in the oceans off of the continental slope, right, the ecology changes dramatically. Perhaps some of the most extreme environments or harshest environments are something like what you're seeing here. which is a black smoker or hydrothermal vent. It's exploding these sulfide minerals that become the source for chemosymbiotic bacteria, right? And so how do you end up with life in some place like this, right? How does something become adapted to deep sea life? If we kind of consider contemporary distributions, right, and modern day taxa in a phylogenetic context, we can examine how a clade might move into the deep sea. So if blue is shallow and red is deep, one way a clade might move into the deep sea is to have a subclade invade and then subsequently diversify within the deep sea. Another way something might happen or something might move deep is to have multiple independent invasions of the deep sea. So an ideal kind of study system to answer a question like this is going to be something that has a broad bathymetric range, right, obviously, but also something that's like worldwide, like globally spread. There's kind of two bivalve taxa that fit this bill. The first is the mittellids, so the mussels, you eat these, right? It's the same clade that you're used to. And then the second are the lucinids, which are the hatchet shell clams. They tend to be found in these, like embedded into these, muddy sands, right? Versus the mittellids form these big beds on rocky substrate, right? When it comes to kind of like comparing two taxa, it doesn't really get, better or more similar than mittellids and lucinids, kind of coincidentally. They have really similar species richness. They're also really similarly aged, according to the fossil record, which is really, really good for bivalves. And they're really, no pun intended, like really deeply divergent, right? They are divided by like 450 million years. These clades have survived all 5 major mass extinction events that have occurred in the world, right? And so there's no like issue of phylogenetic signal like being confused here, right? In this phylogeny, this phylogeny like comes out of our lab, and it's good for a family level, right? But we don't know species level relationships all that well in bivalves. So I went about kind of making a molecular tree with publicly available data for both the mittellids and the lucinids. So I'm not going to go into details over how I built these alignments. There's a lot of code and decisions that go into which sequences to select and whatnot. But just know that in general, these are our stats in terms of the number of genes and base pairs, and I use IQ-tree to infer these trees. And then the other kind of major component of this study is an extensive literature search of the biogeography and the depth ranges of not just the sampled mittellids and lucinids, but every single mitellid and lucinid that exists, right? And so these numbers or these percentages that you're seeing have to do with our sample taxa. You can see it's pretty even for the mittellids and lucinids, especially when it comes to the percentage of shallow water species in these two clades. But then when we consider all mittellids and all lucinids, we can see that, again, these are really similar kind of distributions among shallow and deep waters for these two families. To kind of orient you, when I refer to shallow, I'm saying it is the minimum depth of the species is on the continental shelf, right? Or sorry, yeah, on the continental shelf, so it's less than 200 meters. If it is exclusively below 200 meters, it's going to be deep, right? So it doesn't get any shallower than that. And then in some cases, I'm dividing things into 3 kind of categories, and so there's a both. And so that means its minimum depth is on the continental shelf, but it's also kind of making well way into kind of the continental slope or below. So if we look at our mitellid phylogeny that we've inferred, we can see there's kind of 2 major kind of transitions from shallow to deep within the mittellids. And then there's also kind of in situ diversification below the continental shelf after moving into the deep sea. So when we consider our kind of 2 different diversification scenarios, it's really the first that the mittellids are doing. One of these clades that's doing that is Dacrydium. It looks like there's only like one tip that's deep there. We actually have 2 Dacrydium sampled, and they're sister to each other, and one is like in that both category, which is why it's showing up as blue. But Dacrydium it's like 30 species, and they are kind of mostly deep sea, and we don't know whether or not they're chemosymbiotic, so these are kind of like our weirdos of they probably are, to be honest, but there's no evidence of that right now. And then the real kind of well-known deep sea mittellids or mussels are the Bathymodiolines. They really diversify into the deep sea. If you see a picture, if you like Google search deep sea mussel, it's going to be a Bathymodioline that you see. And And they actually do like all sorts of wacky stuff. They like burrow into or attach to like sunken whale bones and things. And yeah, they are chemosymbiotic. So upon entry to the deep sea, these Bathmobialians are becoming chemosymbiotic in the Middle-Ids. If we if we simulate stochastic character maps based on 1,000 simulated trees, we can see or confirm what we're seeing in the phylogeny, which is that you have a couple shallow to deep transitions, and then you simulate some deep to shallow transitions in reverse, right, or more of them. But that's really just what is occurring within the Bathmobialians, and it also is a matter of how we're dividing deep versus shallow, right, in terms of it's really these taxa are just inching their way back onto the continental shelf, but they're still found deep. In contrast, with the lucinids, we're seeing a lot of transitions into the deep sea. And there are these relatively small phylogenetically isolated deep sea invasions. And so when we're considering our two scenarios, the lucinids are really doing the second one. And so if we look at our stochastic character maps, again, from the simulated trees, we see a lot more transitions from shallow to deep, so a lot more of the pinks, right, in the lucinids than we do in the Middle-Ids. These are to scale over here, so you can just see that there's like 15, or what is it? Yeah, 15 transitions versus 2, right? And so it kind of begs this question of what is driving, or what is allowing lucinids to repeatedly invade the deep sea? How are they different from Middle-Ids in some way, right? Are lucinids maybe, this is kind of a scary term to use, but like, are they pre-adapted to the deep sea in some way, right? Yeah, I don't want to get called out on that, but are they primed to the deep sea somehow? Right? So there's a few possibilities that this might be the case. First, we know that lucinids are chemosymbiotic. It might be a matter of thermal conservatism. So are the putative ancestors of lucinids found in colder or higher latitude waters, right? And then the deep sea is cold, right? So are they getting used to that? And then last but not least are the putative ancestors of the deep sea lucinids. Do they have broader bathymetric ranges, right? And then they're just kind of contracting into these more narrow ranges in the deep sea. So in terms of the first scenario, right, we already know that lucinids are all chemosymbiotic. From the fossil record, we can confirm that they've been chemosymbiotic since the Ordovician. Whereas, like I said, in Middle-Ids, certainly they're not historically chemosymbiotic. The Bathmobialians are, but it's not something that's known to all of them. And but one thing that's like weird about this, potential reason for lucinids diversifying in the deep sea, or their repeated invasions, is the fact that there's no in situ diversification of these lucinid taxa that are in the deep sea in the same way that we're seeing in the Bathmobialians, let's say, or Dacrydium, right? They're not like exploding in the deep sea, they're just going in a lot, right? And so like chemosymbiosis might be helping these repeated invasions, but it's not totally explaining why some lucinids are invading and not the others, right? So let's consider other scenarios. One is, thermal conservatism. Are the, deep sea ancestors of lucinids occupying higher latitudes? And to do that, or to examine that, we can look at, are the present-day sister lineages occupying higher latitudes, right? If we look at the phylogeny again, we have, the blue and the reds and the grays in terms of shallow, deep, and both. And if we highlight the sister taxa, and then throw on their biogeographic ranges. Sorry, I didn't realize this was going to be so small for you guys to be able to see. But if you throw on their biogeographic ranges, where yellow is tropical, teal is extratropical, and gray, again, is both. When we look at just the closest relatives of the deep sea lucinids, they're largely tropical. In general, a lot of the lucinids are tropical, so I don't know how much that's really telling you, right? About like 27% are found in colder water, so either temperate either extratropical or like both, right? And that's not really all that different from the remaining not sister to deep sea lucinids, right? So the sister taxa of deep sea lucinids aren't necessarily doing anything different from the rest of the lucinids. That said, our kind of preliminary search of the fossil record, right, or fossil evidence is suggesting that the deep sea taxa did, and their sisters, so both of them did originate in extratropical waters. So there is that. So the third kind of final kind of possibility for why lucinids are really mobile is, did their ancestors have broader bathymetric ranges? again, if we look at our tree and highlight the sister taxa, right now I only have arrows in this case pointing to the ones that are the sister taxa that are found in both shallow and deep water. So the ones that are kind of passing the continental slope, right? They have the, quote, broader ranges, right? In terms of just like numbers, right, in these categorical variables, it doesn't seem like much, right? It's only 7 out of the 33 sister species that are in both shallow and deep waters. And also the fossil evidence is suggesting that the deep sea lucinids and their sisters are starting shallow, right? So they're not actually, it doesn't seem like they're having these like broader ranges, right? At least from the fossil record. However, if we look at things in terms of non-categorical variables and we just actually look at the maximum depths of these taxa, of these species, when you look at the taxa that are sister to deep sea lucinids versus the rest, their distributions are rather different. It doesn't really seem, it's not really easy to tell, but there is a fatter tail on the more yellow ones, right, the sister taxa. And a two-sample KS test suggests that these are differently shaped distributions. If we just kind of pull out the stats for the sister versus non-sister taxa, we can see that there are proportionally more species with lower maximum depths among the sister taxa than there are among the rest of Lucinidae, right? So they are more likely to be going into the deeper waters past the continental shelf. So yeah, to conclude, there's very different kind of paths to the deep sea, and pre-adaptation or priming, I guess, might be playing a role in that. In the Middleids, we can see a single or two invasions of the deep sea and then diversification within, with one origination of chemosymbiosis captured at the entry to the deep sea. Whereas in the Lucinids, we're seeing multiple small independent invasions. The presence of chemosymbiosis is occurring at the beginning of the clade's origin. And then also, Lucinids might have been primed to the deep sea based on their ancestral bathymetric ranges, which were broader. So they were pre-adapted to maybe colder waters as well. So with that, I'd like to thank everyone here, and then we have I guess a minute for questions. Have anything? Yeah.
Yeah, sorry. I just wasn't clear on how do we know that the Lucinids didn't invade and then diversify? Like, how do we tell the directionality of them, diversifying each other and then invading? Which is the opposite.
You mean, well, they start out shallow based on the fossil record. Is that what you mean? Yeah. Yeah.
So I'm curious, what is the representation of pre-Cretaceous deep sea fossils? Because I imagine you have extremely limited deep sea records, so you should probably be seeing a complete bias towards shallow.
Absolutely. I mean, yeah, there's a bias in the fossil record, right? You're not going to see as many tropical records, and you're also not going to see as many deep sea records. So yeah, things absolutely might be deeper sooner, and we just don't know, right? Yeah, it's a fair point. Cool. Thank you, guys. Thanks.
So I'm going to talk about, if this works, hopefully I'm going to be able to talk to you about adaptive radiation in frogs. So first, I want to acknowledge the people who actually did the heavy lifting on this, which is Álvaro Dugo Cota and Dante Jimenez, which were PhD students in my group. So as we've seen throughout the symposium, one of the main endeavors of evolutionary biologists is really to explain the factors that are responsible for the unequal distribution of species. So what you can see in the map is basically it's a heat map where warm colors depict species richness. So this geographical species gradient was first described by Alexander von Humboldt even before Darwin, and we're still very interested in it. It occurs throughout vertebrate species, so amphibians are no exception to the rule. So most of the amphibian diversity is found in the American tropical region. So about 50% of all amphibians are found in the American tropics. Paradoxically, it is precisely in this region where we have sort of least information about the factors that are driving diversification. And some authors have proposed that one mechanism that can explain this variation in patterns of diversification of species richness across the globe could be adaptive radiation. Some have even referred to it as a phenotype that may account for the entire diversity of life. I think that's a bit of an overstatement, but it's still one of the maybe best examples of Darwin's idea of, I can't see it, the principle of divergence. Because of the key role that ecological speciation plays in adaptive radiation, right? So what's driving the diversification is the adaptation to these novel niches to which the species have access. And a particular case of adaptive radiations are these what are called parallel radiations. So what happens is that you have sort of like multiple adaptive radiations that are taking place in different islands or different lakes, or even like top of mountains, maybe in some groups, where you can have independent invasion of similar microhabitats associated with similar adaptations to those microhabitats, as occurs in the Anolis lizards, as you can see in the picture, or the African cichlids from the Rift Lakes. And what's really cool about these radiations is that what you end up having is that you have convergent evolutions. You have these similar phenotypes that occur as a result of adaptation to the similar microhabitats. And that occurs repeatedly across the different islands or lakes. And one case that we've been studying in my group is the Caribbean Eleutherodactylus, so these rain frogs that are called as well. They're really cool little tiny frogs. Most of them are really, really small. They're direct developing, so there's no tadpole stage. And that really helps because they're less dependent on water for their reproduction, right? They're part of the mega diverse group Terrarana, which are also all like direct developing frogs. They're found, Terrarana is found throughout sort of like southern US, throughout all, Mexico, which is part of North America, and Central America and South America. Terrarana is super diverse. They've got like 900 species. They represent about a third of all American tropical diversity. Eleutherodactylus is part of that group. So they're found from southern United States through Mexico and a bit of Central America. But they're also found in the Caribbean, and that's where most of their diversity is found. So about more than 80% of all Eleutherodactylus are found in the Caribbean. So one of the first studies we did was to describe this radiation. So we found that it's a bit of a busy figure, but I'll walk you through it. So the first thing is that the diversity is the result of a single colonization event. We're not sure, like the data are not clear on whether it was to Cuba or Hispaniola, but it's one of the two. And then from there, they radiated to all the different islands. All major islands, except Jamaica, have been colonized more than once. And it's interesting because in Anolis also, the radiation in Jamaica is monophyletic. So what's really cool is that, so the branches, the colors in the branches represent the islands. On the tips are the microhabitats. So they've invaded different microhabitats independently, both within the islands and across the different islands. So all major islands have most of the microhabitats that are found repeatedly. And we found that there's a significant association between their phenotype and the different microhabitats. But what's even more cool is that there's a significant evolutionary convergence in those traits across all the different islands, independently of like what their ancestor was, where the ancestor lived, right, prior to invading that microhabitat. We've also looked at rates of, temporal rates of diversification. I know that can be a bit matter of discussion, but we've used different methods and have found that although there's no variation in the rates of diversification among coeval lineages, but there is a slowdown in diversification with time, as would be predicted by adaptive radiation theory. But what we were more interested in is looking at, what happens to ecological opportunity? There's been a lot of discussion that, adaptive radiation is driven by ecological opportunity and everything. And with my student Dante, he was saying, but it's like these, the Eleutherodactylus, they didn't invade the Caribbean by themselves. There's been 9 other genera that have invaded the Caribbean. It's not so surprising. I mean, Eleutherodactylus has a richness of over 160 species, and even those 9 genera combined get to about 30 species. But we still wanted to see, well, what are the patterns, because some of them actually invaded the Caribbean, apparently, at least from the molecular phylogenies that have been dated. They apparently invaded the Caribbean prior to Eleutherodactylus, so they had that ecological opportunity. So we're interested in looking at whether the patterns were different from Eleutherodactylus than the remaining groups that also colonized the Caribbean islands. And what we did is use this explicitly island biogeography approach developed by Luis Valente and collaborators. And basically it looks at speciation, extinction, colonization, and whether there's an effect of carrying capacity based on like this island biogeography theory, right? So, well, it's not so surprising, the differences in richness also, told everything before, but like Veronica said in her talk, if everything was really, really clear, then we wouldn't need science, right? So sometimes we have to show it. She said it much better than I did. But so what we found first is, so like I said, we're estimating speciation, extinction, colonization events, and whether there's carrying capacity. So not surprisingly, for the colonization rates are really low, and they're not different for other Anurans and Eleutherodactylus. So the islands are hard to colonize, which is not surprising for something that really has low kind of vagility, right? The extinction is really low for both, so that I found quite interesting. And of course, the speciation rates are much, much, much higher for Eleutherodactylus. But what I find really interesting is that also there's like a massive difference in carrying capacity. So it suggests that they're just better at making the most of that ecological opportunity that is available. So it's not only that it's available, it's that the lineages that diversify are the ones that are best able to make the most out of it. So we've been looking now more recently at phenotypic evolution. So we have about, I'm showing 10 different linear measurements. We had collected more, but these are the ones that stood out as being sort of more consistently associated to the different microhabitats where the species are found. And we also included body size as a measure, SVL as a measure of body size. So instead of combining it in one PC axis, which was a bit messy, we decided to use SVL as body size and then use PC2 and PC3. We also analyzed the traits separately, but for some, some model comparisons and stuff, doing it trait by trait is really complicated. So we've done both approaches. What we found is that the rates of phenotypic evolution seem to parallel the rates of diversification. So we have an apparent slowdown in divergence between species. So disparity tends to decrease as the number of species increase. So we measured this following Mahler et al. 2010, where sympatric species were used as a proxy for ecological opportunity, and we estimated phenotypic disparity at each node, and then we found there's a negative correlation for all traits, almost. However, the effect size of sorry, I just can't see the slides here. So the effect size is quite low. As you see, it goes from about 6% to 13%, if my memory serves me right. So it's not like a massive effect, but we didn't really expect to find a really, really strong effect of it, but still. What is it here? Sorry. Oh yeah, we also looked at different rates of phenotypic evolution. So we wanted to see, we competed different models of phenotypic evolution. So the basic Brownian motion model, which doesn't mean that it's like just sort of like, anyways, that also reflects selection. But we also looked at density-dependent effects and the matching competition model of Newsmere et al. And we found that it's very interesting because for body size, Brownian motion fits best. But for all the other traits, so the PC2 and PC3, which we interpret as shape, it can be discussed, but to a certain extent, it reflects a bit shape. And selection acts on individuals. It doesn't act on specific traits, right? So it's a combination of both. And for PC2 and PC3, we found that the density-dependent models are the ones that fit best. So it's in line with the previous results. So as diversity increases, that seems to limit the rate of phenotypic evolution. We didn't find any effect of competition, but we'll touch on that in a little bit. So the take-home message is that the rates of phenotypic evolution seem to match or mirror to a certain extent the rates of diversification, which is what's predicted by theory. There's also, there seems to be an effect of ecological opportunity measured as the number of sympatric species, which I really understand that it's not the best way to do it, but it's really hard. How do you measure ecological opportunity in a kind of really good way, right? That seems to influence the rates of phenotypic evolution, which again is what would be predicted by theory. And we didn't find an effect of competition, but I wouldn't completely rule it out because we do have this model that accounts for sympatric species. And like if we're accounting for sympatric species, it's because we think that they can interact, right? So there must be some effect of competition, maybe not what we would expect as this character displacement and so on resulting from direct competition, but maybe just limiting access to other species. So it is still an effect of competition, right? I'd like to acknowledge my co-authors, again, Dante and Alvaro, who did the heavy lifting, Carles at Estación Biológica de Doñana, Luis Valente and Dan Roboski. This work is impossible without museums. All the curators and everything have been absolutely wonderful, and there's no way we could have done it. We could have done this without that. And also a shout out, please submit your best work to our society journals. We can be here because of the funding that society journals gives, so send us your best work. I'm sorry, systematic biology should be there as well. Sorry to those that are part of SysBio. And yeah, that's it. I don't know if I have time for questions because I can't see the time.